that is the anti-cancer agents. So this is your syllabus of anti-cancer agents. So we are going to cover these classes, that is alkylating agents, antimetabolites, antibiotics, natural products, platinum compounds, histone deacetylase inhibitors, and the tyrosine kinase inhibitors. And these are the different drugs which are covered in these classes of the anti-cancer agents. Okay. So as we have discussed, this is the very most important topic after the cardiovascular diseases or the cardiovascular agents. Okay, these anti-cancer agents carry most of the weightage after cardiovascular agents. So what actually is the cancer is? So the healthy cells, these healthy cells, they are under strict biochemical control for their growth and the differentiation. Right, whatever the healthy cells are there, that healthy cells, they are under strict biochemical control for their growth and the differentiation. The cells, they divide and proliferate under the influence of under the influence of various growth stimulators, and are subject to arrested growth and the apoptosis, that is the cell death. So as we have discussed, that healthy cells, they are under biochemical control, and also they are under control for their differentiation. The cells, what are the cells are there, they divide and proliferate. They divide and proliferate under the influence of the various growth stimulators, and are subject to arrested growth and the cell death. Now, during cancer, these regulatory processes, that is, those regulatory processes which control the cell, that is, they control the cell division and they control the cell proliferation. So, during the cancer, this regulatory process, they gone wrong. That is, the growth of the cell and the division of the cell. It is not under control. Okay, it is not under control, and that's why the definition of the cancer is what? That is the abnormal division of the cell. Abnormal division of the cell, that is called as the cancer. Now this cancer cell, they spread to the other parts of the body through the blood and the lymph system, and this process is called as the metastasis. So this cancer, it is categorized based on the functions, locations of the cells from which they originate. So this cancer is categorized into the 6 different classes, that is the carcinoma, sarcoma, leukemia, lymphoma, myeloma, and the CNS cancer. In that, the carcinoma is what? Carcinoma is the cancer of the skin or in the tissues that line or cover internal organs. Sarcoma is the cancer of the bone, cartilage, fat, muscle, blood vessels, or other connective or the supportive tissue. Leukemia, it is the cancer of the white blood cells and their precursor cells such as the bone marrow cells, causes large number of abnormal blood cells to be produced and enter the blood. Lymphoma, it is the cancer of the immune system. It is the cancer of the immune system. Myeloma is the cancer of the beta cells that produce the antibodies, and it spread through the lymphatic system. CNS cancer is a cancer that begin in the tissues of the brain and the spinal cord. So now, based on chemotherapy, the cancer is classified on the few classes like the antimetabolites, genotoxic drugs, plant-derived inhibitors of mitosis, plant-derived topoisomerase inhibitors, and the other chemotherapy agents. So the first one is the antimetabolites. Antimetabolites are the agents that interfere with the formation of the key biomolecules, including nucleotides, the building blocks of the DNA. Okay, so antimetabolite agent, what they do? These agents interfere with the formation of the key biomolecules, including nucleotides, which is the building blocks of the DNA. So these agents, they block the biosynthesis of these nucleotides like the ATGC, right? Adenosine, then thymine, guanine, cytosine, whatever the nucleotides are there. So these agents, they block the synthesis of these nucleotides, or they interfere. These antimetabolic agent, they interfere in the linking of the nucleotide. Then the next one is the genotoxic drugs. That is, these drugs, they alkylate or intercalate the DNA, causing the loss of its function. So these genotoxic drugs we are going to cover in the next slides. So how the alkylation of DNA takes place, that we are going to cover in the next slides. Then the plant-derived inhibitors of the mitosis. So plant-derived inhibitors of the mitosis, that is, these agents prevent proper cell division by interfering with the cytoskeletal components that enable the cell to divide. Then the plant-derived topoisomerase inhibitors, as we know what actually is the topoisomerase enzyme, what is the role of the topoisomerase enzyme. This is covered in the biochemistry. It is probably mentioned in the Satyanarayan of the biochemistry. So these are the plant-derived topoisomerase inhibitors. So the topoisomerase enzyme, they unwind or relegate the DNA during the replication. And the other chemotherapy agents, these agents inhibit cell division by mechanism that are not covered in the categories which is okay. So other chemotherapy agents, these are the agents which inhibit the cell division by the mechanism which is not covered in the categories which is listed in the above classes. So this is what about the cell cycle. So what actually is the cell cycle is, now the cell cycle, when the cell cycle reproduce, they do so via a very specific game plan known as the cell cycle. The cell division, that is the mitosis, now this phase, M phase, that is the mitosis phase, it kick off the cycle. Okay, so the cell division, that is the mitosis phase, it kicks off the cycle. So the cell cycle, it start from the mitosis phase, or the division of the cell, it start from the mitosis phase. After a period of 30 to 60 minutes, the cell either enter into the resting phase, that is the G0 phase, or the pre-synthetic phase, or the G1 phase. So it takes around 30 to 60 minutes, right? After 30 to 60 minutes, after mitosis, the cell either enter into the G0 phase, which is the resting phase, or it may enter into the G1 phase, also called as the pre-synthetic phase. In this phase, enzyme production occurs in preparation for the nucleic acid synthesis, whatever for the survival of that cell. So the nucleic acid is required for the survival of that cell. So the enzyme production takes place at this phase. Okay, enzyme production for the cell takes place at this G1 phase. Then production of DNA then occurs in S phase, because what is mentioned in the G1 phase, that is preparation for DNA synthesis. That is, in G1 phase, what happened? That is, they are prepared for the DNA synthesis, and the DNA synthesis occur in the S phase. Right? So what happened in G1 phase? That is, the enzyme production takes place for the survival of the cell. It is occur in the G1 phase. In S phase, DNA synthesis takes place at the S phase. Then it lasts up to the 20 hours. Okay, so for the synthesis of DNA, it takes around the 20 hours. Then S phase is followed by a G2 phase, in which RNA, critical proteins, and the mitotic spindle apparatus are generated for the next mitotic phase. Okay, so this is what about this cell cycle. So the cell cycle, it is start from the mitosis phase. Mitosis phase kick off the cell division. Wait for us. So the cell cycle start from the mitosis phase. From mitosis phase, the cell enter into the G0 phase. Over there, it take rest, or it can enter into the G1 phase, where the enzyme synthesis takes place. From G1 phase, it enter into the S phase where the DNA synthesis takes place. From S phase, it then enter into the G2 phase. In G2 phase, RNA, critical proteins, and the mitotic spindle apparatus is generated for the next mitosis phase. This mitosis phase is again divided into the four different phases, but we are not going to cover all these phases as it is not required briefly in our medicinal chemistry syllabus. So just take into the consideration. So there are only five phases: M phase, G0 phase, G1 phase, S phase, and the G2 phase. So for the mitosis, it takes around 30 to 60 minutes. Then from mitosis, it enter into the S phase, so it takes around the 20 hours. That is the DNA synthesis. The DNA synthesis takes around the 20 hours. So this is what about the cell cycle, cancer cell cycle. Now these are the cell specific and the cell cycle non-specific agents. So there are various anti-cancer agents. They either act on the different phases of the cell, and a few of the agents they are not cell cycle specific. So they act by the different mechanisms. So if you check out the drug bleomycin, that bleomycin agent it act on the G2 phase. Then vinblastine, vincristin, paclitaxel they act on the mitosis phase. Alkylating agent, antitumor antibiotics, nitrosoureas, dacarbazine, cisplatin they act on the G0 phase, and these are the agents they act on the S phase. So here what I have described you about the cell cycle, that is what happened in the G0 phase, what happened in the G1 phase, what happened at the S phase. So that theoretical part it is given over here. So just read it out later on. So what happened, that is the cell cycle, it is start from the M phase, that is the mitosis phase, right? The cell cycle or the cell division it kick from the mitosis phase. From mitosis phase, either the cells enter into the G0 phase or where it take rest, or it can enter into the G1 phase. Then from G1 phase it enter into the S phase. From S phase it enter into the G2 phase, and from G2 phase in G2 phase the cell is ready for the mitosis. Okay, so this is what about the cell cycle. So it is mentioned over here in this slide. Now if you check out now for the synthesis of the cell we require some ribonucleotides, we require some nucleotides. So that nucleotides is made up of some bases like the purine bases and the pyrimidine bases, as we studied this part, right? The nucleotide is made up of some bases, that is the purine base and the pyrimidine base. Also the nucleotide is made up of some sugars and the phosphate molecules. So by combining all these components a nucleotide gets synthesized. Okay, so these are the different agents which act on these different phases during the synthesis of the DNA or the during synthesis of the protein. For example, if you check out the drug 5-fluorouracil, this 5-fluorouracil agent it inhibit the DTMP synthesis. Okay, it inhibit the DTMP synthesis. Then the bleomycin agent it either damage DNA or it prevent the repair of the DNA. Then the alkylating agent mitomycin, cisplatin, they cross-link the DNA. Then the camptothecin, doxorubicin, etoposide, these agents they inhibit the topoisomerase 2 enzyme and also inhibit the RNA synthesis. Dactinomycin agent it intercalates in DNA, inhibit topoisomerase 2 and inhibit RNA synthesis. Zinc alkaloids they inhibit the function of the microtubules. Then pentostatin it inhibit the adenosine deaminase. Adenosine deaminase enzyme is required for the synthesis of the ribonucleotides. Then the 6-mercaptopurine and the 6-thioguanine, they also inhibit the purine synthesis and also inhibit the nucleotide interconversions. Methotrexate also inhibit the purine synthesis and inhibit DTMP synthesis. Cytarabine inhibit DNA polymerase enzyme and also inhibit the RNA function. Then the crescent space, they inhibit the deaminase asparagine and also inhibit the protein synthesis. So these are the different drugs and how they act. Okay, so just overlook this flow chart. we are going to study step by step different drugs. So by that you will properly understand that how the drug actually act on the cell cycle. So this is the first class, that is the DNA cross-linking agent. Okay, and this is the first class of the anti-cancer agent, that is the DNA cross-linking agents. They are also called as the alkylators and the organometallics. So the first subclass of the DNA cross-linking agents is the nitrogen mustard. Second subclass is the nitrosoureas. Then the third subclass is the DNA methylators. Then the fourth subclass is the miscellaneous DNA alkylators. And the fifth one, that is the last one, that is organoplatinum complexes. So these are the five different subclasses of the DNA cross-linking agents we are going to study one by one. So in the today's lecture, we are going to cover the first class, that is the nitrogen mustard. Okay. So all these are the subclasses, that is the nitrogen mustard, nitrosoureas, DNA methylators, miscellaneous DNA alkylators, and the organoplatinum complexes. All these are the DNA cross-linking agents. Okay. So the primary target of the DNA cross-linking agent is the actively dividing DNA molecule. Now, to summarize this slide, the role of the DNA cross-linking agent is what? This DNA cross-linking agent or our drugs or our DNA cross-linking drugs, they are the electrophilic agents. Okay, DNA cross-linking agents, they are the electrophilic agents, and they act on the nucleophilic group, which is present on the DNA. They act on the nucleophilic group which is present on the DNA. On DNA, particularly, they act on the N7 of the guanine. That is, there is a structure of the guanine, and in that guanine structure, they particularly act on the nitrogen atom which is present at the 7th position of the guanine. Okay, and by binding to that N7 of the guanine, they alkylate the DNA. These DNA cross-linking agents, such as the nitrogen mustards and the nitrosoureas, they are bifunctional. Bifunctional means what? That is, one molecule of the drug, it binds to two DNA bases, and thereby one drug molecule can alkylate the two DNA bases at a time. Most commonly, the alkylated bases, they are on different DNA molecules, and interstrand DNA cross-linking through two guanine N7 atoms results. Okay, so as they are bifunctional, so they can alkylate two different DNA molecule by binding to two guanine N7 atoms. These DNA cross-linking agents are not cell cycle specific, but they are more toxic to the cells in the late G1 or the S phase of the cycle. So this is what about the DNA cross-linking agent. So what is the mechanism of action of the DNA cross-linking agents? That is, these DNA cross-linking agents, they are the strong electrophilic drugs. They act on the nucleophilic part of the DNA, that is particularly N7 of the guanine, and thereby they alkylate the DNA. These DNA cross-linking agents, they are bifunctional, means what? They can attack the two different DNA bases at the same time. These DNA cross-linking agents, they are not cell cycle specific, means what? They are more toxic to the cells in the late G1 phase or the S phase of the cell cycle. So this is the diagram of the DNA. As we have discussed, this is the DNA strand. This DNA strand made of these bases A, T, G, C, and these are DNA cross-linking agent. They form linkage with these nucleotides and thereby disturb the DNA structure, or thereby they alkylate the DNA strand. Now over here, I draw example of the DNA cross-linking agent. Now the structure which is pink in color, it is our DNA cross-linking agent, and this is the structure of the guanine. Okay, this is the structure of the guanine. As we have discussed in the previous slide, that this DNA cross-linking agent, they alkylate the DNA. Means what? They are bifunctional. What is the meaning of bifunctional? That is, they alkylate the two different DNA strand at the same time by binding to the N7. So this is the N7, nitrogen number 7. Okay, this is the nitrogen number 7 of this guanine. So these DNA cross-linking agents, so one molecule of the DNA cross-linking agent, they at the same time alkylate the two different DNA bases by binding to the N7 of the guanine. So these are the drugs of the nitrogen mustard. So as discussed, today we are going to cover one class of the DNA cross-linking agent, that is the nitrogen mustard, also called as the aziridine mediated alkylators. So the first drug in this is the Mechlorethamine. Second drug is the Mephalan. Third drug is the Chlorambucil. Fourth drug is the Cyclophosphamide. And the fifth drug is the Busulfan. So today we are going to cover these agents. Now this is the mechanism of action of these nitrogen mustards. That is how the nitrogen mustards alkylate the DNA. So if you check out the nitrogen mustards, they contain this group, that is a bis beta haloalkyl amine. Bis beta haloalkyl amine. How the name come? That is, there is a nitrogen, and to this nitrogen, that is, these haloalkyl amine groups are attached. Haloalkyl groups are attached. So this is all about the bis beta haloalkyl amine. Now this bis beta haloalkyl amine, they undergo intramolecular nucleophilic attack. Intramolecular means what? That is within a molecule. So these bis beta haloalkyl amines, they undergo intramolecular nucleophilic attack to form a aziridinium ion. As discussed, these DNA cross-linking agents, they are the highly electrophilic species. Right? We have discussed in the previous slide. These DNA cross-linking agents, they are the highly electrophilic species, and they attack on the nucleophilic group of the DNA. Right? So over here, by the intramolecular nucleophilic attack, this electrophilic species is formed. If you check out, there are two delta plus charge on these two carbons. So this is what the aziridinium ion. So this is the highly electrophilic species. So once the highly electrophilic species is formed, it attack on the nucleophilic group of the DNA. So once the highly electrophilic species is formed, then they attack on the nucleophilic group of the DNA. So, or where they attack, that is the N7 of the guanine, right? So this is a step 2, that is the intermolecular attack. And this is a step 2, that is the intermolecular attack. That is the nucleophilic group, that is the N7 of the guanine. It attack on the highly electrophilic species, that is the aziridinium ion, and thereby the one DNA strand is get alkylated. One DNA strand is get alkylated. So this is the alkylated DNA. Then again there is one more chlorine group. Okay, so that chlorine group is get liberated as it is a base leaving group. So again the aziridinium ion is formed. Whatever the aziridinium is formed, it is same as which is formed in the step 1. But in the step 1, there was a chlorine group, but now there is a alkylated DNA is attached. So as we have discussed that these DNA cross-linking agents, they are the bifunctional. Bifunctional means what? That is one DNA cross-linking agent can alkylate two different DNA structures at the same time. So we alkylate one DNA structure. Now we have to alkylate the another DNA structure. So how it alkylates? So again the chlorine group is removed, ring closure takes place to form the aziridinium ion. Once the highly electrophilic species is formed, that is the aziridinium ion, then again the N7 of the guanine, that is the nucleophilic group, again it attacks on that electrophilic species to form the uh, cross-linked DNA. That is again this DNA cross-linking agent, that is our agent, it alkylate another DNA molecule to form the cross-linking between the DNA structure. How cross-linking? Because it alkylate one uh, nucleotide by means what this structure, as it is a bifunction, means what it alkylate the two DNA. So if the drug alkylate the two nucleotides which is present in the DNA, then the cross-linking takes place. So the DNA which is formed, it is a cross-linked DNA. Okay. So and later on the two DNA molecule is get liberated from this structure, but whatever the DNA which is liberated from this structure, they are the damaged DNA, they are the alkylated DNA. So if they are the damaged DNA, means what, they are the alkylated DNA, so they cannot harm, or they cannot invade the other cell, they cannot invade, or it cannot spread the cancer to the other cells because they are the damaged DNA. Now all these nitrogen mustard, they decomposed easily when they come in the contact with the water. Now if you check out, now this is the structure of our active mustard, that is the nitrogen mustard. So these nitrogen mustard, when they come in contact with the water molecule, so this chlorine group, which is a base leaving group, it is get liberated, and the OH group is get attached over here, results in the formation of the inactive dehalogenated diol. So this is what about the decomposition of the nitrogen mustards. Okay, so these nitrogen mustards, which is the active drug used in the treatment of the cancer, so they decomposed in the presence of the water. So there are two pathways, pathway A and the pathway B. Pathway A and pathway B. So what is the pathway B? That is the two molecules of the chloride, that is these living groups are liberated, water is get hydroxyl group directly attached, results in the formation of the inactive dehalogenated diol. And the A pathway is what? That is a rearrangement, that is the formation of the aziridinium ion. So it becomes the electrophilic species. Once it becomes electrophilic, then the nucleophilic group, that is the water molecule, it attacked on that electrophilic aziridinium ion. It act on that electrophilic aziridinium ion. Then this is what is formed. Uh, that is the hydroxy group is attached at the one end of this DNA cross-linking agent. Then again one chlorine group is there. So that chlorine group is get liberated. Then aziridinium ion is formed. Once aziridinium ion is formed, then the second hydroxy group, that is the second molecule of water, it attack on the second. It act on that DNA cross-linking agent, and again the inactive dehydrogenated diol is formed. So the first drug of our DNA cross-linking agent, or the first drug of the nitrogen mustard, is the Mechlorethamine. So if you check out the structure, it is same. The R group is over here, it is a methyl group. Okay. So the Mechlorethamine, the IUPAC nomenclature of the Mechlorethamine is bis. What is the general name? That is a bis beta halo alkyl amine. Bis beta halo alkyl amine. So over here the name is bis 2-chloroethyl methyl amine. Okay, that's what the IP nomenclature of the Mechlorethamine. So Mechlorethamine is the only aliphatic nitrogen mustard currently available in the US market. Its use is limited by extremely high reactivity, which leads to rapid and non-specific alkylation of cellular nucleophiles and excessive toxicity. This mechlorethamine is marketed in the salt form and provide water solubility for intravenous and intracavitary administration. This agent is too reactive for oral administration and too toxic to use alone. Treatment: it is used in the Hodgkin's disease, chronic myelocytic leukemia, chronic lymphocytic leukemia. And this is the inactivation of the mechlorethamine by the sodium thiosulfate. So as we have discussed about the decomposition of the nitrogen mustard by the water, so in that slide we have covered that the chlorine group is replaced by the OH group, right? Because the water is the nucleophilic, that nucleophilic group attack on the highly electrophilic species of the nitrogen mustard. Over here, the nucleophilic group is the thiosulfate group. That thiosulfate group replaces the chlorine atom in the nitrogen mustard to form the inactive thiosulfate ester. So this is how the mechlorethamine is get inactivated by the sodium thiosulfate. Second agent is the Mephalan. So the mechlorethamine is the only aliphatic nitrogen mustard, and the remaining nitrogen mustard, they are the aromatic compounds. So Mephalan, it is the first aromatic mustard which is used primarily in the treatment of the multiple myeloma. Okay, Mephalan is orally active, but its absorption can be erratic, and this Mephalan drug is used in the treatment of the multiple myeloma. Now we will check out the IPC nomenclature. Now, if you check out, there is a carboxylic acid. So as per the rules of the IPC nomenclature, we have to give numbering to the structure to the highest priority group, that is the carboxylic acid group. So there are 3 carbons in a straight chain. So 1, 2, 3. But there is a amino group. So we have to follow the our alphabetical order. So the first we have to cover the amino group. Okay, so we'll start our IPC nomenclature from the second position of this propanoic acid. So 2 amino, after 2 amino 3 dash, 1, 2, 3, 3 dash, then 4 dash of this phenyl ring, then after 4 dash bis 2 chloroethyl amino, then phenyl, and at last propanoic acid. So this is what about the Mephalan. Synthesis of Mephalan we will cover later on. Then Chlorambucil. Chlorambucil is also a phenylacetic acid mustard. It is also a aromatic mustard, aromatic nitrogen mustard. This drug has good oral bioavailability and the potential to induce the non-lymphocytic leukemia. Now, regarding the IPC nomenclature, again there are 4 carbons in a straight chain of this acid. So it is a butanoic acid. So there is no any amino group. So we have to cover only this substituent. So 4 dash of this butanoic acid, then again 4 dash of this phenyl, then bis 2 chloroethyl amino, phenyl, and at last butanoic acid. That is all about the chlorambucil. This chlorambucil is used in the palliative treatment of the chronic lymphocytic leukemia, malignant lymphoma, and the Hodgkin disease. And this is the metabolite of the chlorambucil, the acidic metabolite of the chlorambucil. Then the next drug is the cyclophosphamide. The next drug is the cyclophosphamide. Now I think you are aware about this ring. We have covered this ring in the previous chapter in the cardiovascular diseases. The name of the ring is the phosphinine ring, right? Oxazophosphinine. 132 oxazophosphinine. Alright. So what is the IPC nomenclature? That is NN bis 2 chloroethyl, because there is only one functional group, that is the amine functional group. So we will cover this functional group at last. So we'll start with this NN bis 2 chloroethyl, then 132 oxazophosphinine, 2 amine and 2 oxide. 2 amine, 2 oxide. This is all about the cyclophosphamide. This cyclophosphamide is a chiral prodrug anti-neoplastic agent and require activation by metabolic and the non-metabolic processes. So this is the metabolism of the cyclophosphamide. Cyclophosphamide is metabolized into the carbinolamine by the isoenzyme CYP2B6 or the CYP3A4. Also, the cyclophosphamide directly metabolized into its inactive metabolites by removing chloroacetaldehyde by the isoenzyme CYP3A4, or here the N-dechloroethylation takes place. By the N-dechloroethylation, that is the removal of this group to form this inactive metabolite. Also, the carbinolamine, which is synthesized from the cyclophosphamide by the isoenzyme CYP2B6 and the CYP3A4, it undergo hydrolysis. That is, the opening of the ring takes place and results in the formation of the metabolite which contain the aldehyde group, and hence the name is the aldophosphamide. Okay, all the part is same, only the ring is get opened and this alcoholic group is metabolized into its aldehyde form, and the name is what? Aldophosphamide, which is transported into the cell. Then this aldophosphamide is directly metabolized into the phosphoramide mustard. It is decomposed into the phosphoramide mustard. During this decomposition, it liberates a toxic molecule called as the acrolein. It is a uro and nephrotoxic molecule called as the acrolein. So during the metabolism of the aldophosphamide into the phosphoramide mustard, it liberates a toxic molecule called as the acrolein. So this phosphoramide mustard, it undergoes cyclization to form a quaternary aziridinium ion, which is the major product of the cyclophosphamide. And also this phosphoramide mustard further undergo decomposition to form a protonated aziridine by forming few metabolites, that is a bis beta chloroethylamine and the tertiary aziridine free base to form finally a protonated aziridine. So as discussed, the cyclophosphamide is an inactive prodrug and it is metabolized into these several metabolites, that is the carbinolamine, aldophosphamide, phosphoramide mustard, bis beta chloroethylamine, then tertiary aziridine free base, then protonated aziridine, and the quaternary aziridinium ion. These are the different metabolites of the cyclophosphamide. So remember one thing, that is during a metabolism of the cyclophosphamide, a toxic drug is formed, that is the acrolein, which is the uro and the nephrotoxic. The name is the acrolein. So over here again it is mentioned, that is during the metabolism of the cyclophosphamide, few inactive metabolites are formed and the few toxic metabolites are formed. So our toxic metabolite is the acrolein. So this acrolein, it damages the nephron, particularly when used in the high doses. So if we consume a high dose of the cyclophosphamide, then it causes the toxicity to the nephron by the release of the acrolein. In children, when it causes the nephrotoxicity, that is when it is used in the high doses in children, in patients with only one kidney, or when co-administered with the other nephrotoxic agent, that is the cisplatin. So by this time, this drug, cyclophosphamide drug causes the nephrotoxicity, either when it is given in high doses, or when it is given in children, or when it is given in patients who is carrying only one kidney, or also when this cyclophosphamide drug is co-administered with the other nephrotoxic agent. Example is the cisplatin. So to minimize the risk of the bladder toxicity from the acrolein, the fluid should be forced and bladder irrigated. Now the mesna is available as an adjuvant therapy in case of the overtoxicity or as a prophylactic protectorate. A sulfhydryl reagent mesna, it is transported into the bloodstream as the inactive oxidized disulfide and selectively reduced to the reactive sulfhydryl in the proximal tubule. Once excreted into the bladder, the reactive sulfhydryl group, it competes with the system residue for the alkylity acrolein. So in short, if the toxicity occurs because of the use of the acrolein, then at that time the mesna agent is given to overcome the toxicity of the acrolein. Treatment: the cyclophosphamide is commonly used in combination with the other antineoplastic agent to treat a wide range of the neoplasms, including the leukemias and the malignant lymphomas, multiple myeloma. Then also it is used in the breast cancer. This is how the Mesna adduct is used to treat the nephrotoxicity which is caused by the cyclophosphamide. How the nephrotoxicity occur by the use of the cyclophosphamide, that is, during the metabolism of the cyclophosphamide, acrolein is get released, and that acrolein is a nephrotoxic agent. So to overcome this toxicity, this Mesna adduct is used. And the last agent is the Busulfan. The last agent is the Busulfan. Now, if you check out the structure, so the IPC nomenclature is the 4-methyl. If we start from this carbon, so 1, 2, 3, 4. So 4-methyl sulfonyl oxy butyl, and the last portion, that is a methane sulfonate. This one is the sulfonate, methane sulfonate. So these are the Busulfan. Busulfan is an alternative to utilizing aziridines as an electrophilic species. It was found that simply utilizing a carbon chain terminated at both ends by leaving groups gave compounds capable of acting as a cross-linking agents. So in the previous structure, there was a leaving group was chlorine. In this, the leaving group is the sulfonate. Okay. Busulfan utilizes the two sulfonate functionalities as leaving groups, which is separated by 4 carbon chains which react with the DNA to primarily form intra-strand crosslink at the N7 of the guanine. The sulfonate are also subject to displacement by the sulfhydryl functions found in the cysteine and the glutathione, and metabolic products are formed as a result of the nucleophilic attack by this group to generate sulfonium species along with the methane sulfonamic acid. This is followed by conversion to tetrahydrothiophene, and further oxidation products are subsequently produced to give the sulfoxide and the sulfone. The cyclic sulfone, known as sulfolane, may be further oxidized to give 3-hydroxysulfolane. So this is the metabolism of the Busulfan. As the Busulfan is metabolized into this tetrahydrothiophene 1 oxide, then sulfolane, and at last 3-hydroxysulfolane. So these are the different metabolic products of the Busulfan. And how the Busulfan alkylate the DNA, it is shown over here. As we have discussed the mechanism of action of this DNA cross-linking agent. So similarly, Busulfan agents act on these DNA base pairs, right? Means what, the first in the first step, the first sulfone group is released, and the DNA is get attached to that drug, and then later on the another sulfone group is released to attach the second DNA molecule, and this is how the DNA molecule is get alkylated, and this phase is around the metabolic phase of this Busulfan.